My name is Amanda Alker, and I'm a fifth-year PhD candidate in the Shikuma Lab at San Diego State University. I'm here today to talk to you about a project and a question that I've been thinking about for the majority of my PhD, and that is, how can bacteria influence animal metamorphosis? Now, when we think of the word metamorphosis, we may think about butterflies. And when I think about metamorphosis, I think about it in the context of the seafloor. Now, many bottom-dwelling marine animals, like sea urchins, corals, and tube worms, undergo a life history transition similar to butterflies, in that they release their gametes into the water column. They must then develop there, and then form a larvae that swims in the water column for multiple days before it is mature enough or competent to find its place on the seafloor. This process is irreversible, so making the decision to find a good place the first time is very important. In our lab, we study how the larvae make this decision and what factors can play into this. When we zoom in to this precise location where a larvae could end up and eventually become an adult, we find that bacteria can play a role in this. And not just any bacteria, but specific bacteria. Now, it's been shown that bacteria can interact with the larvae by producing a cue. This cue could be a number of different things. And over time, what we've found is that there are generally 3 classes of cues that bacteria can produce that facilitate metamorphosis or that influence the larvae to find its place on the seafloor. These 3 classes are proteins, and an example of that are MACs, or metamorphosis-associated contractile structures, and MACs influence tube-worm metamorphosis. And then there are chemicals. The second class of molecules are chemicals that can influence metamorphosis. And an example of this is tetrabromoperol, or TBP for short, and TBP can influence coral metamorphosis. The last class of molecules that can influence metamorphosis is a little bit more unknown. That is more generally referred to as bacterial components. And what I mean by components is the lipids and the sugars that make up bacterial membranes, which have been shown they may be able to induce metamorphosis as well. And we call this LPS, or lipopolysaccharide, or EPS, exopolysaccharide. So the majority of the first bit of my PhD has been to figure out which of the different types of bacteria that are on the seafloor can produce which cues. And then the second part of my PhD has been to understand a little bit more about how these different classes of molecules can affect different organisms. So that would be induce which organisms, or induce which animals. This is important because we need to know a little bit more of how different animals respond to different classes of cues so that we can harness the bacterial cues that come from very specific bacteria that facilitate metamorphosis to either increase the number of corals that we want to settle down on the seafloor because corals are endangered, or perhaps instead to diminish the number of larvae that are recruited by tube worms, because tube worms are more of a nuisance in a marina. With that, I thank you for your time and have a good day.